So guys, just going to go through the specifications that you need for paper one, and here they are. Cell biology, organization, infection and response, and bioenergetics. Now I've talked to you about the fact that if you know your specs, you are in a massively advantageous position because you are able to look at questions and kind of work out what it might be about. All right. If you don't know your specs, you don't know what you're meant to learn. And when you look at a question, you're not too sure what it's about. You've got no clues. So cell biology, we're talking about eukaryotes and prokaryotes. Eukaryotes have a nucleus. They are the animal, the plant, the fungi and the protists. And the prokaryotes are the bacteria. They don't have a nucleus. They have just a piece of circular DNA and plasmids. And then we need to know about the differences between animal, plant, and bacteria cells. We need to know about the features they have, and therefore working out what they might be. So, for example, if we know that the mitochondria is the site of aerobic respiration, which releases energy, and they ask you what particular organelle, which is a subpart of a cell, a sperm might have, then you'd say mitochondria because it needs energy for swimming. Okay. We then, in our cell biology, talk about mitosis and cell division. We also need to know about microscopy, about the equation that links the actual size, the magnified image, and the magnification, that triangle. We need to know about stem cells, and we need to know, there's a little section, we need to know about the use of stem cells, which we should know, okay, in therapy and medicine, and the fears of stem cells. We then come to organization, and here we are talking about the cells, tissues, organs, organ systems, organism, that hierarchy which you might be asked to put in order. We will be asked specifically about organ systems of the digestive system and the blood, the heart, and the blood vessels. And then we would move on to enzymes, and we would make sure that we know about lock and key hypothesis, enzyme, substrate, key terms of active site, denaturing, and within that we would have the core practicals of food tests and enzyme activity of amylase in the wells when you add starch and see how long it takes to turn blue-black. Okay. Infection and response, we're talking about the pathogens that exist, bacteria, virus, fungi, protists, and examples of those that you need to know. We're talking about the barrier defenses of our bodies, and then the immune response, the phagocytes, the white blood cells, the antibodies, the antitoxins. Okay, we need to know about vaccination. We need to know about that graph that shows the first exposure, where the graph goes up slowly with antibodies on the y-axis and then comes down once the infection's been fought off. And upon a second exposure, the graph, you can imagine it, is higher and steeper. So that means that the antibody response is fast and greater. It also tails off slower, and that's because of the memory cells that have been left within the body after that first exposure, and that's the basis of vaccination. We need to know about drug testing. We need to know about the fact that we start off in the laboratory and then we go to clinical trials on humans, and we need to do all those details. We need to know about the 3 individual drugs. They need to know about digitalis from the foxglove, which helps with heart conditions, aspirin from the willow, which uses headaches, and also penicillin from from fungus, which kills bacteria. Okay, so they're the kind of things that we need to know. We also need to know about the comparison between communicable and non-communicable diseases. Non-communicable diseases are ones that cannot be passed on. So cancer, diabetes, the heart disease. And we need to know about the causes of those, the health factors that are risky, not doing much exercise, alcohol, smoking, etc. Diet, high fat diets are often key. Don't just say you need a balanced diet or bad diet, you specify the high fat diet. Okay. And then moving on to communicable, as we've just discussed, they're diseases that can be passed on. They can be passed on through water, through droplet infection, through sexual intercourse, and we need to know about those kind of things. And so that gives us infection and response. And then bioenergetics, we're talking about two specific things: photosynthesis and respiration. With photosynthesis, we need to know the equation. Whenever you've got a photosynthesis equation, I would write the equation just on a scrap of paper, just so you can look at what plants do. It's what plants do. And you then need to know about the limiting factors: carbon dioxide, temperature, mineral ions for chlorophyll, and light intensity. You need to know the three graphs for light intensity, carbon dioxide, and temperature. Remember, the temperature one actually is the same as an enzyme one, where the enzymes denature after 40 degrees. You need to know about the practical of light intensity moving the pondweed further away from a light. And you need to know about the inverse square law, which is if you double the distance of the light away from the pondweed, you quarter the light intensity. On the converse of that, you need to know if you halve the distance and put it closer, you quadruple light intensity. Okay. And lastly, you need to know about how you would advise a farmer in a greenhouse by telling them whether it's worth economically having a little bit extra light intensity or a little bit extra carbon dioxide or more temperature, bearing in mind the graphs. Often as the graphs begin to tail off, it's not worth the extra money to spend on light intensity and carbon dioxide. And with the temperature graph, you may recall that if you increase the temperature beyond 40 degrees, then the enzymes start denaturing, so photosynthesis actually goes down. Okay, and then the last thing that you need to know is the uses of glucose for amino acids to make proteins, cellulose for cell walls, starch, which is a store because it's insoluble and also doesn't have any osmotic effect, for fats and oils. Okay, and they are our uses of glucose. So that's photosynthesis. And respiration, you need to know about, once again, the equation, which is the reverse of the photosynthesis equation. And you need to know about aerobic and anaerobic respiration. You need to be able to compare anaerobic respiration and aerobic respiration. And my advice there is to write out the equation, just state them. Okay, what's the similarities? What are the differences? You need to know that the aerobic respiration happens in the mitochondria, and that as we do increased exercise and we need more energy and therefore more respiration and therefore more oxygen and glucose, our heart rate and our breathing rate goes up. There will be a point at which we cannot do respiration with oxygen, and that's anaerobic respiration. And we need to know anaerobic respiration, both the equations in animals, which form lactic acid, and in yeast and plants, which form ethanol and carbon dioxide. Okay, we then need to know about reading graphs or the respirometer, in which you might put some maggots in a test tube and you would observe whether they produce oxygen or carbon dioxide. Obviously they don't produce oxygen because they're not photosynthesizing. So when you say, why is the pressure in the test tube changed, you'd say it's because of respiration. Okay, where they're using oxygen and producing carbon dioxide. Okay. Last thing on that is metabolism. The liver, which breaks down proteins, it breaks down urea. It's a place where lactic acid is broken down. Lots of metabolic processes. Okay, and bile is also produced. Okay, the metabolic rate is the sum of all of the chemical reactions going on in the body at any given time. And then linking these two particular topics, we've got respiration and photosynthesis at the same time. If you have a plant in the daytime, it is doing both photosynthesis and respiration. Important to remember that. If you look at the concentration of carbon dioxide and oxygen in a room in the nighttime with a plant, then the oxygen levels are going to go down because it's doing aerobic respiration and no photosynthesis, and the carbon dioxide levels are going to go up because it's doing respiration and no photosynthesis. And in the day, when it's doing both, you might expect the photosynthesis to outstrip the respiration, so it would be absorbing carbon dioxide and it would be producing more oxygen. Okay, and so that is our paper one at a glance. One thing I haven't mentioned, which under cell biology is an important mini topic, is the three methods of transport. Diffusion, movement of particles from an air of high concentration to low concentration down a concentration gradient. Osmosis, exactly the same for water concentration, from an area of high water concentration to an area of low water concentration, but through a semi-permeable membrane, that's the third mark, and active transport against a concentration gradient from a low concentration to a high concentration, requiring energy. So that is a summary in 10 minutes of paper 1. That is the kind of knowledge you need to know about the mini topics that you need to know. Listen to this, learn it.